and welcome to this evening's Icon Textile Group presentation, which will be a back to basics discussion of cotton and wool fibres. My name is Hannah Sutherland and I am the social media rep for the Icon Textile Group, and I'm the host for this evening. Just before we begin, I'm going to run through a little bit of housekeeping. So if we can have the next slide, please. So as everyone is on their home Wi-Fi, just please be patient if we have any connectivity problems. We will endeavour to get things back on track as soon as possible. This is being recorded, so if anyone has any problems and has to disappear, eventually it will be available through a link, which we will send out to participants. Please use the chat window to say hi, maybe let us know where you're watching from. It's always great competition to see who's kind of made the most effort at 5 AM or something. If you have any questions for Chris, just the next slide, please. Then there is a Q&A feature at the bottom of the screen, and if you can use that for your questions, that just helps separate the questions from the kind of nice comments and hellos in the chat window. And then finally, next slide, there we go. If you're having any issues viewing the webinar, we usually find that if you go to the top of the screen and hit to window, that will sort out any issues with the speaker or the slideshow being too small. So without any further ado, I would like to welcome Chris Foster and Francis Hartog just to turn on. Oh, I've forgotten the pub quiz, not the pub quiz, the pub session. Sorry, Maria, thank you. Yes, from 8 o'clock this evening, we are going to be having our virtual needle and thread, which is just an informal networking session, also on Zoom. So if you want to come along between 8 and 9, please do. And we can make the link for that available if you haven't logged in the chat. So sorry, now we will have Chris Foster and Francis Hartog join us on screen, please. Hello, thank you very much. And I'm going to hand over to Francis just to kind of introduce Chris's project and him. Thank you very much.
Thanks, Hannah. Good evening, everyone. Well, Chris is an analytical chemist, and his PhD research is based at the Open University. And his task that he's been set is to ascertain if detergent residues remain on textiles after conservation washing, and if they do, whether they have any detrimental effect on the fibres. Chris doesn't have a textile background, so this was all new to him, and due to coronavirus, he's had to do a lot of literature searches while the labs at the Open Uni were closed. And so the groundwork for his PhD required him to familiarise himself with the makeup and ageing properties of both wool and cotton fibres. And it occurred to me that his investigations could be really useful as a back to basics topic for Icon Textile Conservators. So take it away, Chris.
Thanks, Francis. So hi, I'm Chris, and this presentation is called Fibres Back to Basics. If you could go to the next slide, please. So this is kind of like the run through of how the presentation is going to go. I'm going to start by giving a general description of fibres and some general properties. And then I'm going to go on to speak about wool and then cotton specifically. I'm going to discuss each fibre's chemical structure and the way the fibre is organised. Then I'll talk about how the fibres age with time and under different conditions. And then I'll talk about how the fibre interacts with water, as I thought these were key points which would be useful to a conservator. I should point out that, like Francis said, a lot of this is from the literature, but there is going to be some of my own work put in there, and I'll make point to note that out when I do. Next slide, please. Okay, so what are fibres? Textile fibres are mostly made up of polymers. And polymers are essentially long chains of repeating chemical units, which are called monomers. So each of these monomers, as you'll see from figure 1, has the same or similar chemical structure. And this repeating unit is then connected by covalent bonds. And covalent bonds are quite chemically inert, so that means they're quite unreactive. And they are kind of what you would typically think of in something like a water molecule. So that's H2O, and that's oxygen bound to 2 hydrogen atoms, and that's through covalent bonds. Now, these polymer chains are then connected to other polymer chains by intermolecular bonds. And intermolecular bonds is kind of a fancy way of saying between molecules. So the polymers can be classed as the molecule, and it's the bonds between the molecules. These can range in their bond strength, and this just depends on which type of intermolecular bond can happen. So firstly, you can get van der Waals interactions, and these are fairly weak interactions, and they can actually occur between any two atoms or molecules, as long as they're in close proximity. Then next, much stronger, we have hydrogen bonds, and this obviously needs hydrogen to occur, and also some electronegative atom like nitrogen, oxygen, or fluorine. And then even stronger than this, sometimes in polymers, you can get ionic bonds. And this is electrostatic interaction between two localized charges. So if you think of something like table salt, sodium chloride, that's a sodium atom, which is positively charged, and a chloride ion, which is negatively charged. Again, stronger than this is covalent bonds. So as I mentioned before, the actual polymer is held together by covalent bonds, but different polymer chains can also be held together with covalent bonds. And this is usually called cross-linking, and this is really strong. So these polymers, which are bound through intermolecular bonds, are then organized into these larger structures called fibrils. And if you look in figure 2, you can imagine these long fiber-like, thread-like images as being the fibrils, and fibrils will actually pass through different regions. Some, as you can see from kind of like the hashed region, these areas are tightly packed and have high order, and they're called crystalline regions. The other regions are much more disordered and they have loose packing. These are called amorphous regions. If you could go to the next slide, please. So the ratio between these crystalline and amorphous regions actually determines a property called crystallinity. And usually a polymer that has a high crystallinity will be stiff and inflexible. And this is also seen with the textile as well. Crystallinity can sometimes be linked to the fibre strength, and a way to measure fibre strength is tensile strength testing. And this can be seen in figure 3. You have the textile or fibre clamped between two grips, one which is fixed into place and one which moves at a constant rate of elongation. And we can actually look at the point which the textile or the fibre breaks, and this is noted as the tensile strength. Now, this strength can depend on many different things, including polymer length, the crystallinity, intermolecular bonds, and the actual structure of the fibre itself. Now, another important fibre property to mention is the degree of polymerization, sometimes referred to as DP. This is simply the number of repeating units or monomers in one polymer chain. This is usually an average because in most kind of textiles, the polymer chains will differ slightly in the repeating units per chain. The last property I thought was important to mention was relaxation and shrinkage. When textiles are made through the process, if they're woven or knitted, they're usually put under some kind of stress or tension. And when this occurs, the textile can then remain in that stressed position through forming intermolecular bonds in that position. When water is absorbed, the textile can actually relax as the intermolecular bonds holding the textile in place are broken. And this effect can then lead to a shrinkage, because once the textile dries and the moisture content decreases, if the textile is remained unrestrained whilst drying, it can actually shrink to smaller sizes than the starting dimensions. And this is usually this effect is usually seen more for knitted textiles than woven textiles. Next slide, please. So now moving away from fibres generally, and I want to focus my attention on wool. So wool, as you'll know, is an animal textile fibre, and animal fibres are usually made up of proteins. Now, a protein is formed from types of polymers called polypeptide chains, and these polypeptide chains are made up of subunits called amino acids. as you can see from figure 5. If you're unfamiliar with this type of structure, you can see the atoms labelled, and then at a point where there's an intersection without an atom labelled, that's actually a carbon atom is there. So the amino acid has kind of 3 different regions. It's got the amine group, which is the blue nitrogen area. It has the carboxylic acid group, which is the group that has the 2 oxygens. And then in the centre, it has this R side group, and this side chain will actually determine which amino acid it is. So each amino acid has a slightly different R chain, and these amino acids then form long chains called polypeptide chains, and they usually have a specific sequence of the order of the amino acid. The amino acids present can then determine some of the intermolecular bonds occurring, and these intermolecular bonds can then determine the overall structure of the protein. Now, wool is mostly made up of a protein called alpha keratin. I'm sure most of you have heard of it before because it's what makes up human hair. Alpha keratin contains polypeptide chains which are held in helices, and you can see a helix, which is the singular, in figure 6b. So if you just focus on the red spiral in figure 6b, that's a singular helix. So 2 of these helices will coil together to form a structure known as a coiled coil, which is what is represented in figure 6. A and B are slightly different representations, where A is a space filling diagram, so it's just putting the atoms into space. Now, these coiled coils are held together by different intermolecular bonds. They're held together by weak van der Waals bonds, some stronger ionic bonds, and then we also get these disulfide bonds occurring. Now, these disulfide bonds are a type of covalent bond, which are very strong. And these can occur across the chain itself or between 2 chains. Next slide, please. So these coiled coils of the alpha keratin protein will then form larger structures called microfibrils. These microfibrils have like a rod-like structure, and these again will combine to form even larger structures called macrofibrils. Now, the macrofibril seen in figure 6, which is in the green, is made up mostly of alpha keratin. It's been estimated to be about 90%, although I have seen different figures given in the literature. The remaining portion of the microfibril is made up of a matrix of disordered proteins. So that's kind of a mass of disordered protein. Now, these macrofibrils are then contained in cells within the fibre. If you look at the cross-sectional diagram, which is figure 8, so as if we've taken the fibre and cut it in half and then look down inside. So the cortex is the region with the orthocortical cells and paracortical cells. And this holds the macrofibrils in these cell-like structures. The 2 regions are slightly different, with the orthocortex being slightly more crystalline and the paracortex being slightly more amorphous. And this duality is actually responsible for the curly-like structure that you can find wool naturally when it's like on a sheep. This cortex runs through the whole of the fibre, and this provides a lot of the structure for the fibre too. Surrounding this cortex is this area called the cuticle, and the cuticle has this scaly-like appearance, and it's actually made up of several different layers. I won't go into detail of the different layers now, but just be aware that the epicuticle, which is the most outer layer, has been debated in the literature to the existence, especially in the earlier literature, because many of the techniques weren't sensitive enough to find this region. So if we go on to the next slide, please. So this scaly-like structure, the cuticle that surrounds the fibre, it actually protects the fibre from water entering and also from chemical attack. Now, this happens because the protein here is in a slightly different conformation. It's still keratin, but it's called beta keratin. And the shape of the protein here means that sulfur atoms are actually projected out on the surface. And these are bound to a lipid layer through a thioester bond, which you can see in the figure to the right. And a lipid layer is essentially a fatty layer. It's seen here, and it has this very hydrophobic character. And hydrophobic literally just means it repels water. It's the same as if you put a drop of oil into water, that oil stays in the drop because it's hydrophobic. And so this layer is responsible for that waterproofing of the fibre, and it is also responsible for some of the protection from chemical attack. The cuticle here also actually provides some mechanical strength and structure to the fibre, because the protein is very sulfur rich and therefore it experiences lots of cross-linking through disulfide bonds. Next slide, please. So moving on from the structure of wool and the fine details, I'm going to start talking about how wool ages over time. So wool can be aged in many different ways, through light, heat, humidity, microorganisms, pollution. But one thing that we see is as wool ages with time, it becomes yellow in colour. You can see this in figure 10. These are two of my own samples. They're both wool, Nuns veiling, which is a plain weave. And as you can see on the left, we have an unaged sample of wool. And on the right, we have a sample that's been aged for 12 days at 100 degrees Celsius. These specific values were chosen because Ahmed et al. Had suggested it being equivalent to 100 years of natural aging. One thing I do want to do in my research is see the validity of this statement. So we know that wool becomes yellow with aging, but there's been lots of different reactions suggested to be responsible for this yellowing colour. Some theories suggest an involvement of the amino acid residues, so that's the side chains in the amino acids, and others suggest the destruction of the lipid layer. One thing that we do know is that oxygen is involved in the process. It's an oxidation process of some sort. And this can be seen because if you carry out aging in the absence of oxygen, this yellowing doesn't occur. One reaction which has been put down to be responsible for this yellowing is the breakdown of disulfide bonds. Now, the breakdown of these bonds can be seen in figure 11. This is a mechanistic pathway, and it shows the reaction of keratin with light and oxygen. So in the first part of the reaction, you can see alpha keratin, and I've just, for simplicity, labelled this as two spiral-like structures bound through some disulfide bonds. Now, when this reacts with oxygen and light, where light is the energy source for the reaction, it's kind of the driver for the reaction, we get this product known as cysteine monoxide. Now, this you can see has the disulfide bridge where one of the sulfurs is double bonded to an oxygen atom. This can further react to create cysteine dioxide, and this is where a sulfur atom is bound to two oxygen atoms through double bonds. The final step, which is in fact many different steps, actually breaks the disulfide bond and causes this product called cysteic acid. Now, I've just represented it as SR for ease, but as you can see in the bottom right-hand corner, this SR is actually the sulfur that's bound through the amino acid to the protein. And then it also has two oxygen atoms bonded with also a hydroxyl group, which is this OH group, an alcoholic group. And this is significant because, as I mentioned earlier, disulfide bonds are really strong and provide some structure for the keratin. In reality, I think the yellowing of wool, the reaction that is responsible depends on the aging, and it's probably a combination of all of them. Next slide, please. So the reason that this disulfide bond reaction has been researched a lot in the literature is because it can be really easily tracked using a fairly simple technique called infrared spectroscopy, otherwise known as FTIR spectroscopy. Now, this technique works by using a laser, an infrared laser, and firing it at the sample. You can see in figure 12, there's an image of a sample in the apparatus that I use. This is my own sample. Once you fire the laser at the sample, molecular vibrations can occur. So the actual bonds can vibrate, well, specific bonds can vibrate in certain manners. Then using a special detector, we can tell which regions of the spectrum vibrations are occurring in. So if you look to figure 13, this is an FTIR spectrum. It's a kind of graph. It's not typical in that most graphs you see have peaks from the bottom, whereas the peaks are actually coming from down below. And depending on where this falls in the spectrum, will correspond to different bonds. And in this region with the arrows labelled, these are the sulfur-oxygen double bonds. They give quite a strong band in the spectrum, so we can track these easily. And as you can see, each cysteine oxidation product, so the cysteine monoxide, dioxide, and the cysteic acid, all give slightly different bands because they're in a slightly different environment. And this can be used then to quantitatively track the amount of product that you have in there. And this is because the intensity of the bands is actually related to the amount of product you have in there. So if you go to the next slide. So I have done this, and this is just some preliminary data. And these graphs show days of aging along the x-axis, where the day corresponds to how long it's been in an oven at 100 degrees Celsius. So I've done 0 days, 3, 6, 9, and 12. And then along the y-axis is relative absorbance, and this is essentially the intensity of the band. So it correlates to the amount of that product there is. So as we see for the cysteine monoxide and dioxide, there's a slight increase followed by a decrease. This seems counterintuitive, but it actually correlates with literature. And the reasoning for this decrease is because these are intermediate products, they're not the end point of the reaction. It reaches a point where there's a maximum concentration, and then it's all being used up then to create the final product. And this coincides with what we find with the cysteic acid band showed at the bottom. And this just has a gradual increase of product, which is what we'd expect. And this is just supporting the literature, and it's showing that there is some aging reaction occurring through thermal aging as well. If you could go to the next slide. So I've talked about some of the chemical changes that occur. So there's that damage to the disulfide bonds. There can also be damage to the lipid layer through another oxidation reaction, and possibly some protein backbone damage. Now, the physical implications of this can be, one, a reduction in the waterproofing of the fibre. I've seen this in my own work. I can do some contact angle measurements. So you put the textile with a drop of water and you use a high-speed camera to measure how that water interacts. And you see that with the aged samples, the water is absorbed very quickly, whereas with the unaged, it kind of stays more on the surface. And this is indicative that the wool is becoming more hydrophilic, and therefore that protective layer is being damaged. You can also see a reduction in the degree of polymerization, which, if you remember, is the length of the chains of the polymers. So the actual polymer itself is being damaged. And then this ties in with the last physical implication, which is the reduction in the tensile strength. So the wool will actually become weaker with aging. Below, you can see some electron micrographs. Electron microscopy is similar to standard optical microscopy, which you may be more familiar with, except it uses electrons rather than light to image the textiles. And this is used because you can get to a much lower magnification and much higher detail. So on the left is another one of my samples. It's unaged wool. And you can see in this image, there's quite a defined cuticle. It's that scaly overlapping surface. And there's also some impurities present because this is an unwashed sample. This is just a raw sample. On the right, you can see an aged sample. This has been aged for 12 days in an oven. You can see the effect is quite drastic. It doesn't have a defined cuticle, which is most likely due to damage to the lipid layer, but also this kind of product on the edge could be that oxidation product from the degradation reactions. You can also see areas where the cuticle is actually getting smaller and the lipid layer is peeling off. If you could go to the next. So moving on from how wool ages, and more specifically to how wool interacts with water, I thought this was important to mention. As in conservation, it may be required to do some wet cleaning and also monitor the humidity. So this cuticle layer, although it provides a waterproofing, there are instances when water can actually enter the pores of wool. For example, at high humidity, or when the wool is immersed in water. You could also get this if there's damage to the cuticle. Now, when water enters the fibre, it can actually bind to hydrophilic groups. So that's those groups that are attracted to water inside the fibre. And it has been shown that as the moisture content of the fibre increases, the actual tensile strength will decrease, so the wool will get weaker. And this has been put down to either separation of the fibres and fibrils or breaking of the bonds that hold the wool together. Another effect that can be seen with wool and water is the felting of wool. When wool is immersed in water, the cuticle can actually expand. This can be seen in the top of figure 19. The cuticle kind of opens up. If then mechanical action is applied, fibres with opposing directions can actually interlock, as seen in figure 19. And this felting effect can cause fibres and the whole textile to shrink, and it can be quite significant. This felting process is increased by heat, acid, alkali, and also it increases with increasing mechanical action. So the more mechanical action you use, say, for example, sponging, the more felting that can occur. Okay, next, please. So moving on, that's kind of a brief description of wool. I'm going to now move on to cotton, which is a very different fibre. Wool is an animal fibre, whereas cotton is a vegetable or plant fibre. And plant fibres are composed mostly of cellulose type 1. For the purpose of this talk, I'm just talking about raw cotton, not mercurized cotton, because that can be a slightly different cellulose structure. And so when I mention cellulose in this talk, I'm talking about cellulose type 1. Now, cellulose is a polymer, and it is made up of these subunits, which are called cellubios, where cellubios is actually 2 glucose units, as you can see the area highlighted in blue on figure 20. And these glucose units are bound through a glycosidic bond, which is a carbon-oxygen-carbon bond, which you can see highlighted in the red dash region. And this glycosidic bond then holds the cellubio subunits together too. This polymer has a linear conformation, so it kind of sits in a straight line. There's no twisting as we saw in keratin, for example. These polymers, these linear polymers, are then held together through weak intermolecular forces and then stronger hydrogen bonds, which you can see in figure 21. The red dashed regions are the intermolecular hydrogen bonds. You can also see some hydrogen bonds occur within the chain itself, which are the black dashed lines. These polymer chains are then held in larger structures called microfibrils and macrofibrils. Next slide, please. So these macrofibrils are then held within the fibre in the cell wall. And surrounding the cell wall is the cuticle. The cuticle isn't scaly like we saw with wool. It's actually got a smooth surface that surrounds the fibre. And this is sometimes called the waxy layer or the cotton waxes. And this waxy layer contains fatty acid, pectins and waxes, which are all fairly hydrophobic. And in a similar way that we saw with wool, it's responsible for protection again from water and chemical attack. The cell walls, this primary and secondary cell wall, contain the macrofibrils, and they actually spiral around the fibre axis. And this spiraling provides some strength for the fibre. The primary cell wall is slightly weaker than the secondary cell wall because the angle at which it spirals around the fibre is greater. This spiraling of the macrofibrils causes the whole fibre to have a kind of twisted conformation, which you'll see in a minute. If these macrofibrils change a direction of the spiral, so they're spiraling this way and then they change to that way, a weak point actually occurs in the cell wall, and the whole fibre, the twist of the fibre, will also change direction. And this will be seen in a minute as well. I should lastly just mention the lumen. The lumen is kind of a hollow structure that travels the length of the fibre, and this occurs from the way that the fibre grows. Next slide, please. So moving away from the structure of cotton and onto how cotton ages. So many different chemical changes can occur during aging. That can depend on the type of aging that occurs, whether it's light, heat, humidity. But I'm just going to mention some of the most studied reactions. So firstly, we have the hydrolysis of the glycosidic bond. If you remember, that glycosidic bond is the carbon-oxygen-carbon bond that actually holds the subunits together. And this reaction causes a breaking of these bonds through a reaction with water. The second to mention is chain scission. This is kind of a random breaking of different bonds within the polymer and across the polymer. Lastly is oxidation. So this is a reaction including oxygen, where the bonds will, some of the bonds will break up and other bonds will be made. These spectra that I've highlighted in figure 23 are from my own work. It just shows two spectra. The one in red is, sorry, not red, the one in orange is 12 days aged wool. The one in blue is unaged wool. You can see that the OH vibration increases with aging, whilst the CO vibration decreases. And this could be indicative of hydrolysis being the dominant reaction in my samples, but oxidation and chain scission could also contribute to this. So next slide, please. So now looking at the physical implications this has, again, here's some electron micrographs. This is at a much lower magnification. On the left, you see the unaged cotton, and if you look closely at some of the individual fibres, you can see a twisting in the cotton fibre. There's also regions where there's a kind of twisting and a loop, and this is indicative of those weak points in the fibre, where that direction of the macrofibril spiral changes. To the right, you can see a 12-day aged cotton sample. This at first looks less dramatic than what we saw for the wool. However, if you look closely, you can see some breakage of the fibre in the bottom right-hand corner. This could be apparent in the unaged sample, but it does look like it's both ends of the fibre there. And even more significantly, you can see these circles in the middle of the micrograph, which show an actual separation in a single fibre itself. And this is due to the breaking of those bonds in that degradation. And this does seem concerning. So if we go to the next slide, please. So moving on from how the fibre appears, I want to talk about how some of the properties change. And first, I'm going to mention crystallinity. Now, the crystallinity of cotton is something which has been proposed to have different effects by different studies. Some studies have shown that with thermal aging, there's an increase in crystallinity. And these studies were infrared and Raman spectroscopy. And this increased crystallinity with aging was put down to those amorphous regions, so the disordered regions, being targeted and chain scission occurring, so that breaking of the bonds. And this was causing realignment of the amorphous fibres. So they're actually becoming more ordered and becoming more crystalline, leading to a more crystalline fibre. Other studies have shown that with cotton degradation, crystallinity actually decreases. These have been based on X-ray studies, and these studies suggested the decreased crystallinity was due to a peeling off mechanism. So if we have the polymer chain, the subunit at the very end of the chain peels off and then it's followed along. And this was put down to occur in the crystalline regions. the overall crystallinity changes with aging, in my opinion, from what I've seen, will depend on many different properties, depending on the type of aging and also the experimental technique used to study the crystallinity. An interesting thing that I read is a study that postulated that the surface of the cotton fibres, so the cellulose near the surface and the cellulose in the interior of the fibre, so the bulk of the fibre, will actually experience slightly different reaction mechanisms for the degradation. And looking back at these studies, some of the experimental techniques used to measure the crystallinity were only actually measuring the surface, and some were measuring the bulk as a whole. And so this could possibly, in my opinion, lead to some experimental differences. And also some could argue that some methods to determine crystallinity are more accurate than others. But overall, what's more important is, again, what physically this means to you as a conservator. So it has been seen that with all of these mechanisms of cotton degradation, there is a decrease in degree of polymerization. This means the chains are getting smaller and the actual polymer is becoming damaged. This also coincides with a decrease in the tensile strength. So the cotton fibres are getting weaker with age. Next slide, please. So now kind of finally, I want to mention how cotton interacts with water. Cotton and water show an interesting relationship. When water is absorbed into cotton fibres, it can actually be beneficial in two ways. The first of these is water will actually increase the mechanical strength of fibres. This is, I should mention, dependent on the amount of amorphous, so that disordered region there is within the fibre, because water will enter the pores and it can bind in these amorphous regions, as shown in figure 26. So the water molecules are highlighted in blue, and they actually take part in something called interchain bonding. So they'll sit in between two of the chains and they'll bond to both of the chains. This provides much more strength to the fibre as a whole and more structure to the fibre. Another impact this interchain water bonding can have is it can actually slow down the depolymerization of cellulose when exposed to light. Now, this is a fancy way of saying the degradation that occurs to the polymer, the breaking down of the polymer as a cause of light. And this happens because when the water molecules are bound in between those chains, there's a reduced freedom of movement in the chains. And when light is causing bonds to break, it's much less likely to occur because there's this reduced freedom of movement. I should point out this effect is only significant when large amounts of water are present, particularly if the fibre is immersed in water. And interestingly, I've been reading about paper conservation and how cellulose paper can be bleached using light, and it's usually immersed in water because it was found that this was protecting the paper, and this is probably due to this same effect. So next page, please. So that was kind of a brief refresher on fibre science. I hope it's been useful and that you've either been refreshed or learnt something new. I just now want to talk about my work as a researcher and where I place myself in the field. So one thing that I've been looking at to do with aging is creating a predictive statistical model for the natural aging of wool and cotton using infrared spectroscopy and hopefully Raman spectroscopy. And this is to look at, to validate the use of artificial aging. So in the field, as most of you all know, we use artificial aging a lot as a model. And I want to see how valid this is by comparing it with natural aging. I'm also looking at wet cleaning studies using the anionic surfactant Hostapon TPHC and the non-ionic surfactant Dihypon LS54. And as Francis mentioned earlier, this is to evaluate whether any residual detergent remains after washing and what the impact this could be. I'm also looking at working on some cross-sectional studies using electron microscopy, and this is to look at if there's any changes to the microstructure when washed and aged. I'm also hoping to do more contact angle measurements, and this is to look at the changes in hydrophilicity when textiles and fibres are washed and aged. And lastly, I'll go on to just do some mechanical strength testing to support my findings. Next page, please. So lastly, my acknowledgements. I would just like to thank my funders, the Arts and Humanities Research Council, for making my project possible, alongside with my collaborator, the V&A Museum. I'd like to thank the Collaborative Doctoral Partnership, which is the consortium that my project belongs to, as they put on a lot of workshops and seminars in the heritage sector. I'd also like to thank my supervisors, Simon, Alex and Francis, for their guidance, and all the lab support staff who've helped me. Last little thing to note is my contact details are here. Make a note if you like, and get in touch with any further questions after the Q&A or any opportunities. Thank you.
Fab, thank you so much, Chris. That was wonderful. That was, yeah, really detailed, and yeah, a lot to get to know and think about. I'm just going to start off by asking kind of what made you kind of go into this particular area of research? So what were you doing before you did the PhD?
Yeah, so before I did my PhD, I did a master's, and it was in something totally different. It was in chemistry, and it was actually looking at kind of cancer therapeutics and designing a kind of light sensor, so something that could diagnose and treat cancer. But I've always had a passion for textiles in particular. I like to make a lot of my own clothes and things like that. And I've also had kind of a split desire between science and also the arts, and I have a creative side. So I actually saw it. It was a studentship that I applied for. And when I saw it, I just knew that it was the perfect decision for me. And I just find that I'm very passionate about this subject. It was something that I'd never considered, and now heritage science is something that I'm really passionate about going into.
Fab. And I know that, so I work at the V&A, and I know that you came in to do a wet clean with Francis. Did you find how did that practical experience, did it change anything that you were looking into, or make you rethink anything when you actually saw, the kind of physical steps in the wet cleaning?
Yeah, it did kind of, it was really important for me to see it and help do it firsthand. And what it made me realise is how the decision that has to be made between the wet cleaning of how clean you want to get the textile, but also then to err on the side of caution. So the piece that was being washed was quite a fine cotton textile, and there was some quite dark staining, and we managed to lift the staining quite considerably, but we had to leave some of it because we were worried that the textile might not cope. Yeah. And it was interesting to see how much of it's half an art and half a science between getting it right. And it was interesting to me because obviously I'm trying to do very specific, detailed, repeatable studies. And it's made me realize that when if I publish or when I produce my thesis, I need to make it clear that I've used models specifically so that I can get repeatable results, but in practice, it's actually not that simple, and a lot of thought has to go into it.
Yeah, it's a little bit of witchcraft involved. Okay, so we've had a couple of questions come through. So we've got one from an anonymous, which just says, thanks Chris for your presentation, really interesting and detailed, pitched well for a non-scientist. I wondered if you would be able to summarise the role of RH, so relative humidity, in your artificial aging experiments.
Yeah, so currently I actually haven't studied the relative humidity, but it's something that I want to go into. I started with just doing thermal because it was the easiest. So in my thermal experiments, they're all kept at the same temperature. They're not done in the absence of light, so light is present, but it's only through a small window and it's all kept controlled. I hope to go on to do some relative humidity, as this is obviously a very important thing to consider, but I think that will be future works.
Ooh, take myself off mute. I'm just going to check the chat for any more. Ksenia has asked if you have any thoughts on the use of deionized and or soft water, tap water, I'm guessing in regards to kind of cleaning textiles.
Yeah, so for my experiments, I'm actually using deionized water, and this is just for sake of reproducibility. But I understand that with anionic surfactants, it's very important to not be using hard water, and this is because it can really interact and make the anionic surfactant less efficient. I know that some conservators use soft, some use deionized, and sometimes it's a case of what's available, but I don't specifically know which one's better. I would personally always just use deionized. I don't know if Frances has any opinions on this.
Oh, she's staying quiet.
Okay.
I'm sure she does.
Hi. Well, I suppose, yeah, I mean, deionized is more expensive is one thing. And also, I mean, the soft water is helpful if you're using an anionic, just because it lifts the pH, and you know, the higher the pH, the more effective the cleaning. So I think that's as much as far as my knowledge goes.
And that's a sensible, yeah, consideration scientifically.
Thank you. We've had another one through which just asks if you can comment on water temperature, and is that something that you look into?
So currently, my studies are just based on a standard water temperature, but I'm aware that water temperature can play a big role, especially if you're using surfactants, and this can be for different reasons. For example, with non-ionic surfactants, they have a cloud point, which means that the surfactant won't be as effective, and with anionic surfactants, oh, has Francis got a hand up? Oh no.
No, keep going.
Oh, okay. And with anionic surfactants, it's also important to get the temperature right. I know that with the felting of wool, that can be increased by heat, but I don't actually know how the heat of the temperature of the water can affect the mechanical strength, but I'm sure it's actually somewhere in the literature.
Okay, but another couple come through. So you mentioned your further research into detergents, into how detergents affect the fibres. Do you have any preliminary data for this, or any expectations of kind of what you might find when you carry that out?
So far, I've been looking at if I can detect any residual detergent on the surface, and I've done kind of two different experiments for both wool and cotton. I have used unaged and aged wool to wash, and I've used very specific concentrations. So firstly, I've used a lower concentration, something more similar to a standard. And then I've also used concentrated. The first technique I used with infrared spectroscopy, and at first it didn't seem that you could detect the surfactant. However, when I then repeated this with the more concentrated, I used kind of very fancy statistics to look at changes in the spectrum that you couldn't necessarily see with your eye. And when I did this, I was getting the exact spectrum for hostapon and dihypon, and it was showing that it was definitely present. And this was a 5 times the standard concentration. I've also done some preliminary contact angle measurements, and these were looking at the change in the hydrophilicity of the surface, and I was seeing that there was a difference for wash samples. I was also, but this was, I'm unsure if this was due to the surfactant or another contaminant that was present. So far I'm still kind of working it out. I expect that there's going to be some residual, because I would be very surprised if, even though the rinsing is very vigorous, I would be surprised if it was completely free of surfactant. But I'm just at the moment trying to find a technique that might be sensitive enough, and then to be able to quantitatively identify how much it is absorbing too.
Great. Another one which just asks about, so you've done your aged testing with your wool and cotton samples. Have you got any historic objects that you are comparing your kind of test subjects to, and how did you pick those objects?
Yeah, so I don't actually have any objects to compare to, but I have been running kind of just a mini experiment where for a long time I've had samples that have all been washed or unwashed, and I've actually placed them in kind of a controlled region in kind of a windowsill. And the idea is to try and emulate if something was on open display, for example, for a short period of time. And the reason I've done this is because me and my supervisors have agreed that we need to be very scientific about this, because I want to get very reproducible results to confidently say this is happening and these reactions or this mechanism is happening. And the reason we're doing this is because with historical objects, we're unsure of the provenance, even if it's been in a museum for a long time. We're unsure what additives have been added. If it's been washed, there may be other additives added to it. And so we're just trying to get a very, very precise statistical method. And the methods I use, the predictive statistics, requires it to be very controlled.
Yeah, there was one more. So this, it might be a similar kind of answer, I guess, when you're having to repeat things to get your kind of scientific testing at a certain level. But Glenn Charnock's just asked, could I ask what rinse methods you were using for the residue testing? You said it was vigorous, but what is your method?
Yeah, okay, so I have a standard procedure. I use, so for the anionic surfactant, I use 2 grams per litre of the anionic surfactant, and then for the dehypon, it's gone from my head, I think it's 1.5 grams per litre, but that could be wrong. I'd have to check my notes. But so I basically use a small kind of wash bath, and I have 2 separate ones that are cleaned, and I'll then I'll first do an initial soak in water for a couple of minutes, and then I will take that out, and then I will use the surfactant, and I'll do an initial surfactant bath where I sponge using forrama sponges. They're like a kind of soft body sponge. And I will then sponge, and I use a specific number of sponging throughout, and I do a 15-minute bath. I'll then rinse in between, and I'll do a running rinse and also a soak in clean deionized water. And then after this, I'll then do another surfactant bath, the same as before. And then after this, I do a running rinse, and I'll do a running rinse for 5 minutes. And then I'll follow this with a soak in deionized water for 10 minutes. And then I use a clean, fresh forrama sponge, and I'll sponge at the beginning and end of the bath for a specific number. And this is just to remove any residual detergent, hopefully. And then I'll repeat this bath method, rinse, bath, of just deionized water. And I'll do that for 4 to 6 times, depending on which surfactant. And the sponging is only done in the first 2 baths, I should say, the extra sponging. And this is, I know this is quite short, a much more reduced kind of time period, as I understand it can take a whole day to do the real thing. But we're hoping that this gives some kind of specific standard.
Yeah, great. And you mentioned the Rama sponges there. How do you prep them beforehand? Do you do anything to them fresh out the packet?
Yeah,
I think you and Francis have had some discussions about this, I know.
Yeah, so I was finding the contact angle measurements tests I mentioned earlier. I was finding there was a difference in the hydrophilicity of the washed and unwashed samples, and this could be expected if residue detergent was there. But I did a control where I washed in water and I used the sponges, and I'd been taking the sponges out of the packet, soaking overnight, and then rinsing and squeezing the sponges and rinsing and squeezing. And when I did this, I had an observation when I was actually washing the samples in water, there was a slight foaming occurring, which is indicative that there's a surfactant there. And this was a bit concerning. And then I looked at the contact angle measurements, and the water wash samples were actually having the same effect as the samples washed in surfactant, which was very worrying to me, as this is not what I would expect. So after a lot of thinking, I put this down to a possible contaminant. So I did actually contact the company and ask, because I noticed there is a kind of residue when you take the sponge out of the packet. And I contacted them, and all they would say is that it's a biocide. And after doing several different analytical techniques, I could see that it's most likely a surfactant. And in my opinion, I think it's a cationic surfactant. As these are quite useful biocides. And obviously this is worrying, as we don't want anything extra on the textile. So now I understand that Francis has been running a washing machine on a cycle, a full cycle, with absolutely nothing in there, just to rinse out the washing machine, and then putting the sponges in the washing machine with nothing in, to try and get rid of all of that detergent that's there. I'm also copying this at the moment to then do some other studies to compare if this is getting rid of all of that surfactant. But it's just something to be aware of. I think if you do plenty of rinsing, and especially at a higher temperature, I wouldn't go too high, maybe like 60, because from memory, I think the sponges can go to about 80 or slightly above. So yeah, if you are using that, you'd want to be careful.
Thank you for that. Well, it's almost our time up. There's no more questions come through. So I just want to say a really big thank you to you and to Francis, and yeah, for everyone watching who wants to listen again. It will be recorded and we will make that available. We're getting some applause through on the chat, Chris. So thank you very much. And if anybody wants to get in touch with Chris, he has kindly given his email. If you missed it earlier, we can share that around. If you email icontextilegroup@gmail.com, we will get that back to you. So I think it's just a good night for now. And if anyone is joining in with the pub, that will be kicking off at 8 p.m. So time to go and get a cup of tea, glass of wine, bit of dinner. Great, thank you very much, everyone. Bye.